Welcome everybody who are going to use BioLabImage program. In this video I would like to demonstrate the functions of this program, and to open the file we have two options. One is to use main menu, file, open, and we can open JPEG file with colonies. What we would like to do here, we would like to detect all colonies on this plate. As you can see, we have two options. One for analysis of gel blots and one for analysis of cells colonies. So we should press this button, and then here on the left side we have different options to color sectors, contours, frames, but now we're interested in this section. This is about colonies. What we have to do, we have to activate the color wheel. To do this, just click this button, then we'll have option to choose the different colors. First we have to select color for colonies. To do this, we just can press on the colony, and then as you can see we have the color detected for this colony, and then we switch to background, activate the color wheel for background. To have the color for background, we just have to click on background, and now we have the color of the background. Next, what we have to do, just press the search and count colonies. If we do this, then we detect single colonies. As you can see, in some cases we didn't detect cells because they are together or have a little bit different color. To count these colonies, what we can do, we can click and drag, and then you see this yellow box, and if we do this, we added one colony to the count, and then we can add more. It doesn't matter if they are crossing each other, just boxes corresponding to each colonies which we would like to add to our count. As you can see, we, I guess, marked all colonies, but we have the false detection. We can again drag box, and then if we double click on this box, it will disappear together with false detection. If we made some mistake and detected wrong colony, we can again double click, let's say here, double click, and then this box is disappear. So we just have to add more, one, and then at the right side in the table we see the number of counts for this plate. If we decided to do this again, we can clean the image just by pressing this button. We can remove all layers again, and then we can repeat procedure. And this function is working for colored images or for grayscale images. If we press this button, we can switch to grayscale mode, and then we can repeat procedure which we did before. If we would like to count in some areas on this plate, we can make the grid. Let's say the grid has size 12 cells on each side. So on this, now we see the grid. We can change the size of this grid. We can change color. Let's say it's red, and so on. We can add text to this image. You see the text here. What we can do, if we just click and drag, then we can change position. Again, just double click to remove text. If let's say we would like to have bigger letters and different color, red one, and then again press and click and drag, then we have our image. A second function is about detection of cells. For this we can open new file, cells. In this case we have tissue, and cells are labeled with antibodies conjugated with different fluorescent dye. So let's say we would like to detect all cells with this color. We select cells and colonies. And in this case, we should use the color wheels for cells. This is here on the bottom. And procedure is the same. We should activate color wheel and press, let's say, this cell. Then activate background and press background. Then we have background here. And then just press search and count. As you can see, now we have detected these cells. We have contours around the cells. We can change color of these contours. So as you can see, we have 95 detected cells of this color. Some of the cells are not detected, and again we can do the same procedure as we did with colonies. We can click and drag and then it will count. One more cell, as you can see. We can repeat the same procedure, but we can change manually the color of cells. For this we have to switch to HSB mode, and then we can change saturation, brightness, and then we can play with these parameters to change the efficiency of detection. What we can do more, we can increase the complexity of the analysis. Let's say we would like to count cells in two areas. What we can do, first we draw a sector, click on this button, and then as you can see, mouse pointer is changed, and then we can outline the sector where we would like to do our analysis. Let's say first sector here, and if we double click it will close, and this sector A, and let's say we have another sector here, sector B. And then we repeat procedure which we did before. To do this we have to switch off this draw sector button, and then we have to activate again cells color wheel and background color wheel, but in our case it's already we set the colors for these two parameters, and then we simply have to press again search button. And if we press, we see in the table on the right side we have two sector A and B, and for each sector we have detected some cells, and outside of the sector we also have cells designated as rest. So this is additional option for detection of cells. So next function of this program is about calculation of band intensity, relative band intensity for gels, blots. And to do this we should press this button. But first of all, let's say we open new window, and then we will not use main menu but drag and drop the image in this area, DNA gel, grayscale. Let's say we need to calculate the intensity of these bands. For this, what we have to do, we should set the background. It's already set automatically as dark, and we have dark background. And then we have to click and drag, and then we have boxes. Actually the same boxes as we have made before for cells detections. Boxes around the bands. In this case it will not play too much role how exact you draw the boxes around the bands, because all this background color will be subtracted for intensity calculations. Then we have to choose numbering for these bands, vertical or horizontal. Let's say horizontal. And finally we should press this button to calculate relative intensity of bands inside the frames. As you can see, in this table we have relative intensities, and in our case the bend intensity in box 2 is the lowest one. We have fold and we have numbers, and if somebody would like to have different reference, not the lowest intensity, then you can use these numbers to recalculate in a way as you wish. Again, we can make some label, just let's say again red, press and put somewhere. As in the case of colonies detection, cells detection, there is option to copy the image which we see on this in this area. Two finger and click, and we can copy this image in the buffer. If we would like to put this image in the keynote, we can pass this image to the keynote. This is it. Thank you for your attention. Bye.